Okay, hello everyone, I'm Nazia, and I'll be presenting a paper on efficient cancer modeling through CRISPR-Cas9 HDR-based somatic precision gene editing in mice. So we all have heard more or less about CRISPR-Cas9 system. However, this system was actually invented quite some time ago, and several new modifications have been made since then. So I'll be talking about one of the latest discoveries that utilized this technology and made it better for cancer cell modeling or tumor modeling. And this is a preclinical test, definitely, because it was carried out on somatic cell line of murine, like murine somatic cell line, or like mice somatic cell line. This paper was taken from Science Advances, and it was published on 2023 May. And here you can see the author names. I have also given the DOI here so that you can check out this paper later on if you want. Okay. So the aim of this study was to improve mouse cancer models using a modified CRISPR-Cas9 system for precision or for precise gene editing. So what is the CRISPR-Cas9 system? We know that CRISPR stands for clustered, regularly interspaced, short palindromic repeats. This system was adopted from a natural defense mechanism in bacteria, where this short sequence was integrated into the bacterial genome by a bacteriophage. And this system can actually eliminate similar bacteriophages when it enters the bacteria's body. So using this kind of like a cutting mechanism, we were able to actually do gene editing. However, there still are several drawbacks of the CRISPR-Cas9 process, because when it starts cutting, it can cut it can cut out a lot of parts of the gene, and it's not that specific. So that's an issue. Like, you can't go into gene editing and end up cutting different fragments randomly, right? Because that might create several problems downstream. So in a CRISPR-Cas9 system, there are two main parts, being the Cas9 protein, which is like an enzyme that cuts, and a guide RNA that guides this protein to a particular location where it needs to be cut. Like on the DNA, excuse me. The Cas9 enzyme also cuts the DNA at targeted location, and cells repair the cut, allowing for insertion, deletion, or alteration of DNA sequences. By alteration here, I mean that, point mutations that aren't necessarily indels. So this is like a brief overview of CRISPR-Cas9, and this system can be used for knocking out genes, or like getting rid of certain genes in the DNA. So coming on to the introduction. So the most common mouse model for cancer study includes genetically engineered models, or GEMMS, and virus-mediated models. So these virus-mediated models actually have some limitations. This GEMMS affects the germ line and doesn't really mimic sporadic human cancers well. So what do I mean by sporadic human cancers? These are cancers that can occur like randomly, right? Like spontaneously. And the drawback of a viral model is that it lacks native gene expression controls, like naturally how a gene is being expressed. It lacks that control. And as a result, they have unpredictable genome integration, and we definitely don't want that. CRISPR-Cas9 allows editing endogenous genes that are already present, right, in our body, in their native locations, overcoming the issues of viral models. So what we are trying to eliminate here is that we are trying to get rid of, like random insertions or unpredictable genome integration that's often seen in viral models. So we are trying to get rid of that. CRISPR can also create indels and point mutations, as I've mentioned before, and base editing and prime editing methods also have some limitations. So coupling this CRISPR method with homology-directed repair, or HDR, can introduce precise point mutations. So far we had CRISPR-Cas9, but now what we're trying to do, or what the scientists try to do, is they have tried to they combine HDR with CRISPR-Cas9 so that they can introduce more precise point mutations. So what's the difference between these two systems? In CRISPR-Cas9, it targets specific DNA segments using a guide RNA, and CRISPR-Cas9 HDR is also kind of similar, since it can also target the cutting process. In CRISPR-Cas9, the Cas9 enzyme cuts the enzyme. So the Cas9 enzyme cuts the DNA, and like in the same way, CRISPR-Cas9 also Cas9 enzyme cuts the DNA. So in CRISPR-Cas9 non-homologous end joining, this is the process DNA, like the default repair mechanism is used. That's normally found inside our human body as well. And this can often, like this NHEJ, can often cause insertions or deletions at the cut site and mainly used for gene disruption. However, the newer model, which is CRISPR-Cas9 HDR, it requires something extra. So what is that? It's a donor DNA template for precise editing. So this donor DNA template is the proper DNA template, like the one that we are trying to, the one that we want in our system mainly, like not the mutated one. It's like the kind of like the wild type DNA, okay, donor DNA template. So based on the donor DNA template, the HDR can insert or correct specific DNA sequences. As a result, this can also be used for fixing mutations instead of just causing gene disruption like the previous model of CRISPR-Cas9. Okay, am I clear so far? Hello?
Yes,
Yes, you're presenting very well. Yes.
Okay, so I can continue. Thank you. Okay, so in vivo editing of proto-oncogenes for cancer modeling faced challenges in efficiency and robustness so far. And this study actually reports a modified CRISPR-Cas9 HDR method for efficient and flexible editing of proto-oncogenes in mouse mammary glands to create tumor models. So this can also be applicable to other cancers as well, but here we have, like here the scientists have chosen mammary glands, or to see how breast cancer is, like, can be modeled. So this new method actually overcomes the limitations of models. So now I'll be moving on to the materials and methods section. Hello, can you hear me?
Yes, you are clear.
Okay.
So
I think I was having some
there were some
internet.
fluctuations. Yes, there was. I thought it was my maybe some technical problem, but I don't know. Maybe
No, I
if
think
there
it's
is a bit
my
time ago. Okay.
yeah,
It's
I think the problem is
unavoidable.
on my
It's
end.
okay.
Yeah. Okay, so I'll be going briefly through the materials and methods. So at first, for plasmid creation, the scientists developed a lenticras G12D. So the mutation was made at the 12th position, and the G became the D amino acid, and lenti peaked to CAH1047R. So this modified AAV KPL plasmid or adenovirus plasmid for specific gene edits. And this process also utilized PCR and restriction enzymes for precise genetic alterations. After that, virus was produced. This virus was actually taken from another university's lab, like Baylor College's lab. And they produced lentivirus and adenoviruses using specified methods for plasmid transfection and virus purification. So actually in the paper, the detailed protocol for this wasn't given. And so I did not really dig that deep in, but if you're interested, you could definitely look up like the reference from the paper. Okay, so transgenic mice and care. So used specific strains of mice bred for desired genetic traits. So transgenic mice was used, like, of specific strains, And they were bred for desired genetic traits, and of course, animal care protocols were followed, and mostly actually female mice were taken in this research because like cells from female mice were taken because females are more prone to develop breast cancer. And intraductal injection was also administered, and inside the injection of obviously the modified biductal injection procedures, it meant that in the mammary duct, this injection was actually given. Okay. Afterwards, mammary gland imaging was made, and using fluorescent microscopy. Then the tissue was processed, like tissue of the mammary glands were processed and stained. So this actually used H&E staining methods, where eosine was the dye. Afterwards, single cell suspensions and flow cytometry was made, and these cell suspensions were made from mammary glands for the flow cytometry analysis. As we know that flow cytometry is a process where the cell numbers and cell sizes can actually be determined using this technique. Then laser capture microdissection was done for precise collection of tumor components from tissue sections. Afterwards, genomic DNA purification was made, where DNA was extracted from tissues and microdissected areas for analysis. Then the edited genomic regions were detected using PCR and sequencing. So here, two types of sequencing were used. However, I believe that deep sequencing is something that I did not hear about before. So what deep sequencing is, it's a type of next generation sequencing, but here particular regions of the genome is actually sequenced multiple times, like maybe hundreds and then thousands of times, to kind of to detect some rare genetic disorders. So the deep sequencing was also used here to identify specific gene edits in the tumor DNA. Finally, off-target survey was done, where genomic DNA was analyzed for unintended edits using specific sequencing and analysis methods. Then RPPA and RNA sequencing was done, which is like a reverse. Excuse me. Excuse me. So RPPA is mainly reverse phase protein array, which can be used to determine protein expression variation. And DNA sequencing was also done to study the gene expression changes. So gene transcript clustering and enrichment analysis was performed, like on the RNA sequencing data. And finally, statistical analysis was done using students' t-test to compare the two methods. You know, the first method being the virus-mediated one, and the second one being the new model. So students' t-test was used for that, and also Kaplan-Meier plots were also used to check like which method is more consistent. So the results were as follows. An AAV vector system edits CRAS in mouse mammary glands and induces tumors with high efficiency. So this was proven. So CRAS is a gene that's responsible for tumor genesis, and specifically for breast cancer. This is an important gene. So to enhance tumor modeling in mammary glands using CRISPR-Cas9 HDR and compare it with previous lentivirus or retrovirus, this method was carried out. So here, an adeno-associated virus or AAV vector was used to avoid permanent gene integration, as seen with retroviral or lentivirus vectors. So what's the difference between a lentivirus vector and adeno-associated virus vectors? My internet connection. Hello, can you hear me?
Yes, you're still connected and we can hear you.
Okay. Thank you. Okay, so in case of adeno-associated virus, the mutation or the gene integration is kind of temporary. Like the viral genome does not get permanently integrated into the host genome. So this is more preferred compared to lentivirus or retrovirus, where the new gene gets integrated permanently. So that's why AAV was used. So AAV serotypes 1 to 9 are known to infect mouse mammary gland epithelial cells. So this was taken from literature review. Then AAV9 carrying COP-GFP was injected into mammary glands to test infection efficiency. That remember the ductal injection. So through that it was introduced into the body of the mice. Then green fluorescence observed in ducts and flow cytometry showed that about 0.4% infection rate, so which is much lower compared to the 4% with lentivirus. So this confirmed that AAV serotype 9 is suitable for infecting mammary gland epithelial cells. So here you can see a picture where AAV serotype 9 infects the mammary gland epithelial cells. So what you can see from here is that this method was actually successful in incorporating the in basically causing tumorigenesis in the mammary gland. Afterwards, an AAV vector system also this CRAS proto-oncogene was used for its frequent mutations in human cancers and previous success in inducing mammary tumors in mice. And the modified AAV vector was mainly used to avoid immunogenic elements like Cas9 activation, Cre and luciferase, and to focus solely on editing CRAS. So this actually enabled this actually prevented immunogenic activation. As a result, the scientists were able to see only the effect of the edited CRAS gene. Then they also demonstrated efficient tumor induction in mammary glands, and that was comparable to previous methods, but different tumor cell differentiation was driven by edited CRAS versus the overexpressed CRAS G12D, the mutation that they actually were interested in. Then they observed the differences in tumor progression and characteristics between AAVK and lenticras G12D induced tumors. And from this comparison, it was seen that the AAVK was a better system than the lenti one. So here, in figure 3, you can see it's given about installing the CRAS G12D mutation through somatic editing efficiency. And that actually induced tumors in mouse mammary glands. So here you can see in the first one, it's given that okay, the HDR allele and the wild type allele is given over here, where you can see the wild type allele was changed in parts, right? So the red portions, they're actually mutations. And in the second one, you can see the Kaplan-Meier graph, where tumor-free survival percentages are given, and days of injection are also given over here. From here, it can be said that the researchers were actually, overall the figure tells us that the researchers successfully introduced a specific oncogenic mutation into the mouse genome. They then monitored the impact of this mutation on the onset of tumor development using the Kaplan-Meier survival analysis, showing that the mutation leads to tumor formation in both general and specific MMTV Wnt1 genetic backgrounds, with the latter being more rapid. And here also, like the Sanger sequencing was also done that confirmed the introduction of the mutation, and the bar graph below, like I don't know how much you can see the bar graph, it's a bit covered, but means occurrence at the genomic level. So, okay. So in this picture, you can see the histology of mammary tumors induced by oncogene lentivirus delivery method and gene editing method. So as I have mentioned during the, while explaining the materials and methods section, the H&E method, that's basically hematoxylin and eosin method. So this was used to stain the mammary tumors, and the virus is used, and the genetic background is also indicated here. For example, in the first one, you can see lenticras G12D, and then lentipic3CAH1047R. So these are the two different types of genes that were studied. And in like A, C, E, and G, for these four, you can see the see what happened after the lentivirus was introduced, and for B, D, F, and H, you can see what happened after the adeno associated virus was introduced. And this also showed that the AAV method was better because it introduced the mutation, or basically the oncogene expression was better in the images shown by the AAV one. So the sequencing also confirmed that the KRAS G12D mutation and synonymous mutations indicating tumors resulted from precise genome editing. Then next generation sequencing showed that about 50% of reads had the KRAS G12D mutation. So this actually validated the editing efficiency of the newer method. Then the AAV-K's ability was tested to edit KRAS in precancerous cells with existing oncogenic alterations. And that also showed that the scientists observed that rapid tumor development was seen in MMTV Wnt1 CAG SP-Cas9 mice, which was basically the, like Wnt1 is, it's like a pathway that helps in the development of cancer. So as a result, this Wnt1 background, or Wnt1 cancerous background, was used to test these two models on. Okay, so rapid tumor development was seen in this background when when the newer model, like CRISPR-Cas9 HDR model, was used compared to the previous method of using like the lentivirus one. So they also noted differences in tumor types between AAVK and lenti1, suggesting that different impacts of edited versus overexpressed KRAS G12D. So AAVK actually induced tumors that closely resemble spontaneous tumors, indicating a more natural evolution than lentivirus-based models. So the last line here is very important. Like that was the whole point of this experiment, right? That like which model can induce more spontaneous tumors, because in our body, tumors or cancer cells, they actually occur spontaneously, right? They don't necessarily occur with set rules or at set times. So mimicking this spontaneity in an in vivo model, or like in an in vitro model, is extremely crucial, because that would help us to associate these systems more closely with human bodies, right? So this method also suggested a more natural method, or natural evolution, compared to the lentivirus-based models. Afterwards, genome-edited PIK3CA induces mammary tumors with high efficiency. This was also proven. So expanded the vector system to edit the PIK3CA gene frequently mutated in human breast cancer. So the developed AAV construct with the PIK3CA gRNA, gRNA means guide RNA, and HDR for the H1047R mutation. So these were developed, this construct was developed, then injected into the mice. After that, tumor formation was seen. with latency similar to lentivirus model. However, the scientists there noticed that a notable histological differences between the tumors was seen in the newer model. So sequencing further confirmed that the intended H1047R mutation and the synonymous changes in tumors. So here also you can see a graph that actually showed the mutation and how it affected the like the tumorigenesis in mammary glands. So this graph is actually similar to a previous graph you have seen. Here you can see that in number B, you can see that like the Kaplan-Meier curve basically shows that the tumor-free survival mice over time after being injected with gene editing tools. So here two groups are compared, right? One receiving a lentivirus and another with AAV vector. So the red line actually shows that AAVP drops more quickly than the black line. This indicated that the mice receiving AAV vector developed tumors faster and more frequently than those receiving the lentiviral vector. So this actually played into, this actually further confirmed that the newer model was better. And in number C, you can see that in this specific genetic background, which probably has a predisposition to developing tumors due to activation of the Wnt pathway, which, as I've mentioned before, is a key signal in cell proliferation and cancer. The tumor-free survival after gene editing is shown here. The mice in this genetic background also develops tumor very rapidly after the injection of the AAV vector carrying the PIK3CH1047R mutation, as indicated by the sharp decline in the red curve. So in D, you can see the Sanger sequencing chromatogram, if I'm not wrong. Yeah. So this chromatogram also verifies the presence of the intended PIK3CA mutation and the top sequence of HDR that actually shows both the wild type and mutant peaks at the mutation site, indicating the HDR process was successful but resulted in a more mosaic pattern. So what do I mean by mosaic pattern? It means that a mix of edited and unedited cells. Nonetheless, it was good enough for the experiment. And finally, in the last graph, the bar graph of editing efficiency, this graph quantified the percentage of reads from sequencing that showed the mutation at each position of the genome that was targeted, and high, these bars actually indicate the percentage of mutations, which also implies higher editing efficiency. And the different color corresponds to the 4 DNA bases, which was inserted at each position during gene editing process. Here you can see mostly these are C's and G's. Yeah. Then genome-edited PIK3CA induces mammary tumors with high efficiency. Approximately 50% of the reads from deep sequencing showed the mutated allele, indicating primarily heterozygous editing of PIK3CA. Then the laser capture microdissection and sequencing of isolated carcinoma confirms the heterozygous PIK3CA editing. Then the whole genome sequencing revealed, so here they're talking about like the different types of methods and what was the purpose of each. So the whole genome sequencing revealed minimal off-target edits, which indicated high specificity of the vector system. So AAVP induced rapid tumor development, as I've mentioned before while explaining the image, is that in the Wnt1 mice, suggesting synergy between P13K and Wnt signaling in mammary tumorigenesis. Then they also observed significant histological differences between tumors induced by AAVP and lenti, indicating diverse impacts on tumor cell differentiation, as I have shown in this diagram. So here you can see the histological differences. So all of this suggested that definitely the second or the newer model was better. Then genome-edited proto-oncogenes can also activate more consistent cellular signaling changes than lentivirus or lentivector-delivered exogenous oncogenes. So virus-delivered mutated genes also show varied expression levels due to integration side effects, right? As I've mentioned in the beginning, like why they are not that good. And that may also lead to diverse oncogenic signaling among cells. CRISPR-edited genes, on the other hand, they remain under native regulatory control, right? Potentially resulting in less tumor heterogeneity compared to lentivirus-induced tumors. Reverse phase protein array, or RPPA, profiling of the mammary tumors actually revealed that no exclusive clustering. So that indicated moderate to high intertumoral heterogeneity across all the models. And on the MMTV Wnt1 background, the CRISPR-edited tumors also showed less heterogeneity than lentivirus-induced tumors. That suggested more consistent signaling perturbations. Perturbations or changes. And faster tumorigenesis on the MMTV Wnt1 background also may limit secondary genetic or epigenetic events affecting the protein profile consistency, or the protein expression mainly. So here also you can see the comparison of tumors generated by the proto-oncogene editing and lentivirus-mediated delivery, where it's again showed that the proto-oncogene editing method was much better than the lentivirus-mediated oncogene delivery. So the genome-edited proto-oncogene can also activate more consistent cellular signaling changes than lentivirus vector-delivered exogenic oncogenes. So to further confirm that, they conducted RNA sequencing analysis on 4 groups of the PIK3CA tumors to assess the intertumoral heterogeneity. And only the tumors with edited PIK3CA on the MMTV Wnt1 background formed a distinct cluster. So this gene editing leads to more uniform tumor formation, particularly on the Wnt1 background. So, okay. So, so this a comparative analysis was also performed between tumors initiated by gene editing and lenti-delivered oncogenes to assess the differences in proteins and signaling pathways. And after using the students' t-test for comparison, no significant differences were found in most comparisons. However, notable differences in protein features between the two systems were found on the MMTV Wnt1 background. So that was what we needed, right? Like on the tumor-induced background, how the effect is. Then differential protein expression, especially in cell differentiation in proteins, may also explain histological differences in tumor types. So oncogenic drivers from both methods can also cause similar or distinct signaling changes affecting the tumor characteristics. So here they have talked about breast cancer, but this can also be applicable to other cancers, and several studies can be done based on that. So the genome editing models also revealed signaling network alteration caused by a Wnt1 transgene better than the lenti oncogene overexpression models. So the CRISPR-edited tumors on the Wnt1 background showed less heterogeneity. So this potentially reflected that the Wnt1 signaling more accurately than lenti oncogene models. Then GSCA analysis also revealed that Wnt signaling pathways in CRISPR-edited PIK3CA tumors, but not in lenti PIK3CA tumors. And the RPPA dataset analysis compared protein features in CRISPR and lenti method-induced tumors, with CRISPR showing more protein feature differences. So this was also a more robust method. The CRISPR method also detected a greater impact of Wnt signaling on protein networks and identified proteins as components of Wnt signaling. This suggested that the CRISPR method's superiority in revealing these pathways was far greater. So here you can see an image that shows the somatic proto-oncogene editing method can better reveal Wnt signaling protein features than the lentivirus oncogene delivery method. So here the Venn diagram also shows the differential protein features from comparing the two different ones, where you can see both of these mutations. So what's the summary of this whole experiment? Let's come to that. So far they have carried out a lot of different types of experiments just to prove that why the newer method is better than the already existing methods. And here, like time and time again, I have talked about the MMTV Wnt1 transgenic mouse model. So what does that even stand for? So MMTV stands for mouse mammary tumor virus that can actually induce mammary tumors in mice, in the context of transgenic mice, of course. And this MMTV promoter is used to drive the expression of genes of interest, specifically in mammary tissue. And Wnt1 is the gene that encodes for a protein in the Wnt signaling pathway that's actually involved in growth, proliferation, differentiation, etc. And dysregulation in this pathway can be seen in different types of cancer, including breast cancer. That's why this particular Wnt1 background was used for this whole study. So the enhanced gene editing in mammary cells achieved high efficiency tumor induction by precisely targeting two key proto-oncogenes, which was so the CRAS and the PIK3CA, demonstrating a leap in modeling accuracy and efficiency. Innovations include circumventing immunogenic elements like Cas9 and ensuring the homology-directed repair domains remains undisturbed for optimal DNA repair and gene editing. So which one was more stable? Like the scientists, they have found that the AAV model was much stable to test this out. This approach also surpassed the older models by avoiding permanent gene integration and maintaining native gene regulation, thereby offering a more physiological representation of cancer. What does this mean? That the native representation of a cancer, like how it naturally occurs in our body, that was maintained, instead of just randomly, permanently inserting certain editing in the genome that can actually have long-term detrimental effects. So this method also provided a closer simulation of human cancer evolution, making it valuable for studying cancer biology and assessing potential treatment options. So the versatility of the technique actually allows for its application across different tissues and animals, broadening the scope of cancer research. So perhaps instead of mouse models, different other models can also be used, like zebrafish or like silkworms maybe. So the study also outlines strategies to target gene editing to specific cell types, enhancing the precision of the model. And the improved model also revealed unique aspects of cancer development and treatment response, uncovering insights not observable in previous models. So this new method can also provide us with unique aspects or like new perspectives on cancer development, like in natural or native state, how the cancer actually develops, that can be studied as well. So to conclude, the CRISPR-Cas9 HDR improvement is basically that basically enhanced the gene editing method to efficiently introduce different methods into somatic cells for mouse tumor models. And this also mimics human cancer by creating a model that's more accurate and more related to human cancer development and useful for studying cancer evolution and therapy responses as well. Like, for example, I believe that this type of method can also be used to test different types of drugs, like which drug better responds to, for example, breast cancer. So multiple drugs can be compared using this type of model maybe. And this study also, like this newer technique also demonstrated unique aspects of cancer evolution in models of two common human proto-oncogenes, and that wasn't normally seen in traditional lentivirus-based models. And maybe more proto-oncogenes, like of different types of cancers, can also be used to like also be tested on these new models. So that's all about my presentation. I know this was a long presentation, and there's actually a lot of depth to this, like different techniques as well. But due to the limitation of time, I did not like mention those. But yeah, of course, if you are interested, you could always read the paper and also go to the references to check out the different types of methods that were used in this study. And I also need to actually study the techniques, because so many of these techniques were very new for me to learn as well. I did not know about a lot of these techniques because this is a very recent paper, right? So the techniques are also quite novel here. So yeah, I shared this paper because this was something new that I learned about, and I hope that this was interesting to you as well. Thank you.
Thank you too. If anyone have any question, it was a tediously long one, but
Yeah.
the result was quite, I mean, not just only important, it could be, like a turn of the event. If anyhow they can know how does cancer cell grow and how they function, the mechanism, and how to inhibit them, there'll be like tremendous victory in every field anyway. So if anyone have any question regarding on the paper she presented, that would actually clear up lots of confusion. Have any questions and any term you don't understand. So far we have Arifa Rahman Shairi. Hello, Arifa.
Hello, Miss.
Hi, you can call me. Actually, I actually don't say my name. It's Shampriti, so don't call me Miss. I'm also a student like you, so my name is fine. Don't call me Miss.
ওকে ওকে আমার কোনো কোয়েশ্চেন নাই, আমার কাছেও এই টপিকটা নতুন। আমিও এই ইয়ে থেকে প্রেজেন্টেশন থেকে আমি জানতে পারছি অনেক কিছু, বাট ফর নাও আমার কোনো কোয়েশ্চেন নাই। আর অনেক জায়গায় আমি মিস করে গেছি কারণ আমারও নেট প্রবলেম ছিল, আর আপুরও মেবি ইয়ে ছিল দেখে আমি অনেক কিছু মিস করছি। তো যখন নাকি রেকর্ডিং দেখবো তখন মেবি কিছু ইয়ে করলে আমি বলতে
হ্যাঁ,
পারবো।
আপনার কোনো
এখন
কোশ্চেন, হ্যাঁ, কোনো কোশ্চেন থাকলে তখন আপনি ধরেন হচ্ছে গ্রুপে শেয়ার করলেন, আপনাকে মেনশন করে দিলেন, বা যে কোনো কেউ থাকলে ডিসকাশনও করা যায়। এটা আরো ভালো হয়, ডাইনামিকটা ঠিক
Yeah,
থাকে,
sure,
না
we can
হলে
do that. Like আমিও এই
yes.
টপিকটা নিয়ে আসলে এখন শিখছি, কারণ এটা আসলে মানে নতুন একটা রিসার্চ, তো এটা মানে অনেক নতুন টেকনিক্স ইউজ করা হয়েছে যেগুলো একটু ডিফিকাল্ট আসলে বোঝা। লাইক আমারও অনেক জায়গায় আসলে গ্যাপস আছে, সো আরো ইন ডিটেল স্টাডি করতে হবে। So
ইভেন
yeah,
ধরেন আপনাদের
level
যদি
should be great.
এটা কিন্তু এরকম না যে এখানে আপনার একটা প্রতিষ্ঠানের সাথে আসছেন, এখানে কাজ করলেন চলে গেলেন, এখানে আপনাদের নেটওয়ার্কিং হয়। এখানে যারা যারা আসছে সবাই রিসার্চের প্রতি ইন্টারেস্টেড। তো দেখা গেলো আপনারা হইতেছে আমাদের মাধ্যমে না হলেও আপনারা নিজেরা একটা স্টাডি করতে পারেন, রিসার্চ করতে পারেন, বা ধরেন হচ্ছে আমাদের মাধ্যমে করলেও আমরা একটা জুম মিটিং করতে পারেন, আমরা না থাকি মানে আপনারা আপনাদের একটা গ্রুপ স্টাডি করতে পারেন, নিজেরাও করতে পারেন, আমাদেরকে বললে আমরা সেটা অ্যারেঞ্জ করে দিতে পারি। আমার মনে হয় এটা করা হয়তো আপনাদের জন্য ইম্পর্টেন্ট। আচ্ছা, তাসনিম তাসিন, হ্যালো, sorry, তাসনিমা তাসিন, are you there or okay? আমি হ্যালো, yes, অ্যাকচুয়ালি আপনার ভয়েস অনেক লো, মানে অনেক আস্তে শোনা যাচ্ছে। And now yes, it's better now. আচ্ছা, ব্যাপার হচ্ছে আজকে আমার ফার্স্ট টাইম এই প্রেজেন্টেশনে হচ্ছে জয়েন করা, শোনা। আমার জন্য টপিকটা পুরাই নতুন, ওই কারণে আমি আসলে কোনো কিছু দিয়ে কন্ট্রিবিউট করতে পারছি না, বা কোনো
আচ্ছা,
প্রেজেন্টেশনে
সমস্যা নেই।
হতে
আপনার
পারছি
কোনো
না।
ধরুন, হ্যাঁ, আমাদের সবার কাছে এটা অনেক অনেক কিছু টার্ম নতুন, অ্যাডভান্স, হয়তো আগে কোনো শুনিয়ে নাই। সো মানে বেসিক কোনো জিনিস যেটা হইতেছে আপনি যদি সামারিটাও যদি না বুঝতে পারেন বা আপনার কোনো কনফিউশন থাকে, আপনি আমরা ডিসকাস করতে পারি, আপু ডিসকাস করতে পারে। আজকের জন্য না, প্রবাবলি
in future আমি আর বেশি
ওকে
এগিয়ে
শিওর।
যেতে পারবো।
হ্যাঁ, আপনি পেপারটা পড়তেও পারেন, এটা যেহেতু শেয়ার করা আছে অনেকগুলো সাইটেই, গ্রুপে, তো আপনি যেখান থেকে আপনি কানেক্টেড ওইখান থেকে আপনি কালেক্ট করে নিতে পারবেন। ওকে,
Sure,
থ্যাংক ইউ।
alright. Thank you.
ওকে, অ্যান্ড স্বর্ণা রেজা,
Hello.
হাই।
Um, Actually, I didn't join from the very beginning, but I have a silly question. That is, why we use mice for the cancer modeling over other animal, or we see that many of these experiments mouse is used. Why we choose mouse over other animal for experiments?
Okay, to answer this question, actually mice model is so popular. Like one of the main reason is that it's first of all it's a mammal and it's very closely related to human beings, right? And yeah, then you can say that okay, there are also other mammals, so why aren't they used? So mice is particularly used because the way a disease develops inside the body of a mouse is very similar to that of a human being. And it's also quite easy to grow and take care of mice compared to some other animals. And higher organisms, or like, for example, monkey or orangutan, which are more similarly related to human, those are not used because the ethical issues over there is larger. So like by comparing all the pros and cons, like mice is like the most suitable one because of their physiology, because of their behavioral patterns, and they also have like a similar like brain and like what do you call that, like brain composition like humans as well. Like they are also quite intelligent creatures. So yeah, as a result, like after studying all the pros and cons, mice are selected for like a proper model organism, like one of the model organisms to use to study. There are also other model organisms,
Okay.
like a fruit fly, or what's that called? Canorhabditis elegans. I forgot the proper, like the actual name of that. But yeah, other model organisms are also used.
I did CRISPR-Cas9 method few years ago.
Yes.
I have please explain CRISPR and Cas9 method. মানে explain this. আমি এটা হচ্ছে মানে অনেকদিন আগে পড়েছিলাম। আমি শুরু থেকে জয়েন করিনি, তো মানে আমি একটু এটা রিভিউ করতে চাচ্ছি মেথডটা। মেথড বলতে পুরো মেথড না, মানে এই জিনিসটা আমি একটু জানতে চাচ্ছি।
আচ্ছা, হ্যাঁ, শিওর। সো লাইক এই পেপারটা হচ্ছে মেইনলি CRISPR-Cas9 মেথডটার আরো মডিফাইড একটা ভার্সন ওরা স্টাডি করেছে, যেখানে হচ্ছে CRISPR-Cas9 মেথডটাকে আর কি HDR মেথডের সাথে ওরা লাইক কম্বাইন করেছে। সো CRISPR mainly stands for so CRISPR mainly stands for clustered repeats of interspace short palindromic like repeats, right? So এটা হচ্ছে মেইনলি CRISPR-Cas9। আর Cas9 হচ্ছে একটা এনজাইম যেটা হচ্ছে ডিএনএর বিভিন্ন পার্টে কাট করার জন্য ইউজ করা হতে পারে। সো CRISPR-Cas9 মেথডটা হচ্ছে মেইনলি ব্যাকটেরিয়ার মধ্যে পাওয়া যায়, যেটা হচ্ছে একটা ব্যাকটেরিয়াকে যখন একটা ভাইরাস ইনফেক্ট করে, ওই ভাইরাসটাকে আমরা ব্যাকটেরিওফেজ বলি, রাইট? সো ওই ভাইরাসটা যখন ইনফেক্ট করে তখন ওই ভাইরাসটাকে কিভাবে ব্যাকটেরিয়াটা বডির ভিতর থেকে বের করে দেয় বা ভাইরাসটাকে মেরে ফেলে। সো এই মারার মেথডটাই হচ্ছে মেইনলি CRISPR-Cas9 মেথড। সো এটা কিভাবে মারতে পারে, বিকজ ব্যাকটেরিয়া নিজের বডির মধ্যেও ভাইরাসের একটা ছোট পোর্শন, এই যে লাইক একটা ছোট ডিএনএ সিকোয়েন্স হচ্ছে মানে ইন্টিগ্রেটেড হয়ে গিয়েছে, অ্যাজ আ রেজাল্ট ওটা রেকগনাইজ করতে পারে, ভাইরাসটাকে রেকগনাইজ করে পরে মেরে ফেলে, রাইট? সো ক্রিসপার
আচ্ছা,
ক্যাস9
এটা
এর আইডিয়াটা
জিন
ওখান থেকেই নেওয়া হয়েছে।
একটা মেথড না, জিন এডিট করে
জি,
এটা এটা দিয়ে জিন এডিট করা হয় না?
হ্যাঁ হ্যাঁ। সো এই ব্যাকটেরিয়ার এই মেথডটা দেখার পরে সায়েন্টিস্টদের মাথায় বুদ্ধি আসলো যে আচ্ছা এটা তো ইউজ করে জিন এডিট করা যেতে পারে, রাইট? সো জিনটাকে কিভাবে এডিট করে, যে জিনের কিছু পোর্শন থেকে ইন্ডেল করা যায়, মানে বেস ইনসার্টও করা যায়, বেস ডিলিটও করা যায়, বা পয়েন্ট মিউটেশন করা যায়, মানে একটা একটা বেসকে আর একটা বেসে চেঞ্জ করা যেতে পারে। সো এসবের জন্য ক্রিসপার ক্যাস9টা ইউজ করা হয়। হাউএভার এই মেথডটার কিছু ড্রব্যাকস আছে। সো ড্রব্যাকস এর আগে বলি, যেটা মেইন দুইটা পার্ট থাকে, একটা হচ্ছে ক্যাস9 প্রোটিন, যেটা হচ্ছে এনজাইমটা যেটা কাট করে, আর একটা হচ্ছে গাইড আরএনএ, যেটা হচ্ছে কোথায় কাট করতে হবে ওইটা বলে, ঠিক আছে? বাট এই মেথডটা হচ্ছে খুব একটা বেশি প্রমিসিং না, কারণ হচ্ছে এটা র্যান্ডম জায়গায় জিন কাট করে ফেলে। সো র্যান্ডমলি যে জায়গায় যদি জিন কাট করা শুরু করে তখন তো এটা প্রবলেম্যাটিক, এটা জিন ডিসরাপশন করতে পারে। ইটস গুড ফর ডিলিটিং আ জিন বা নকিং আউট আ জিন, দ্যাটস ফাইন, বাট একটা পার্টের মিউটেশনকে ফিক্স করার জন্য এটা অতটা ভালো না। এইটার কারণে এই ক্রিসপার ক্যাস9 মেথডটাকে এইচডিআর এর সাথে কম্বাইন করা হয়। সো এইচডিআর কি? ওটা হচ্ছে হোমোলজি ডিরেক্টেড রিপেয়ার। সো এইটা হচ্ছে আমাদের মানে নর্মালি বডিতে কি নন হোমোলোগাস এন্ড জয়নিং রিপেয়ার একটা প্যাথওয়ে আছে, ডিফল্ট যেটা রিপেয়ার মেকানিজম। সো ওইটার পরিবর্তে এটা হচ্ছে কি করে, যে হোমোলজি ডিরেক্টেড আর কি রিপেয়ারটা করে, যেটা সো এই মেথডের মধ্যে একটা ডোনার ডিএনএ টেমপ্লেটও ইউজ করা হয়। সো ডোনার ডিএনএ টেমপ্লেটটা কি? ওটা হচ্ছে একটা প্রপার লাইক ওয়াইল্ড টাইপ ডিএনএ টেমপ্লেট, যেটা ডিএনএ সিকোয়েন্সে কোনো মিউটেশন বা কিছু নাই, যেটা আমরা চাই। সো এই ডিএনএটাকেও হচ্ছে এই সিস্টেমের মধ্যে ঢুকানো হয়, পরে ওইটার উপর বেস করে মিউটেশন গুলো ফিক্স করা হয়, ঠিক আছে? সো এই এইচডিআরটাই ইনসার্ট করে এই স্পেসিফিক ডিএনএ সিকোয়েন্স গুলো কারেক্ট করে ফেলে। সো তাই ক্রিসপার ক্যাস9, যেটা পুরানো একটা মেথড, এটার চাইতে হচ্ছে ক্রিসপার ক্যাস9 এইচডিআর মেথডটা আরো বেশি হেল্পফুল। তো এইটা কেন হেল্পফুল, এটার কারণেই হচ্ছে বিভিন্ন ধরনের এরা এক্সপেরিমেন্ট করলো এই মাউস মডেলের উপর, এই ব্রেস্ট ক্যান্সারের একটা টিউমার মডেল ডেভেলপ করার জন্য, করে প্রমাণ করলো যে আচ্ছা এই মেথডটা আসলে অনেক সুইটেবল, এটা বিভিন্ন ধরনের ক্যান্সার স্টাডিতে আসলে অনেক ইজিলি ইউজ করা যেতে পারে, এটা থেরাপিউটিক্স ডিসকভারিতে ইউজ করা যেতে পারে, এটার অনেক ওয়াইড রেঞ্জ অফ অ্যাপ্লিকেশন হতে পারে।
আচ্ছা, থ্যাংক ইউ, বুঝতে পেরেছি।
ওকে, ইউ ওয়েলকাম।
ওকে, নাজিয়া, নাজরুল, নাফসি, আপনার প্রেজেন্টেশন খুবই ভালো ছিল, and আপনার এক্সপ্লেনেশনও ভালো ছিল। So overall, I think it's a good presentation. Paper choosing was right, and Sir couldn't join us. I guess he is busy.
ইয়েস স্যার, আগের বারও থাকতে পারেননি আমার
হ্যাঁ,
প্রেজেন্টেশনের সময়।
but I think when he get time, he will check this presentation. I will recommend because this was a good one, and we can also keep it as a standard. You know, it was a good one. We can show people that you can learn from this presentation how to present. Anyway, and I guess
I
Nadia,
wish
you
I
are
could
preparing
like
for
yeah, I wish I could
preparing
improve
for
for pages and make the writing shorter though. আমি আসলে মানে খুব একটা বেশিটা করতে পারিনি, সেজন্য
no,
একটু
you
long
were
হয়ে
sick,
গিয়েছে
so you did a very
প্রেজেন্টেশনটা।
good job in that. Actually, আপনি sick থাকার পরও হচ্ছে attend
আমি
করছেন,
আসলে
join
এডিট
করছেন.
করতে
That's
পারিনি।
a lot. No, that's actually the motivation. অনেকেই আছে যারা হইতেছে completely fine, but they are not presenting. They do have time, but not presenting. So it's very encouraging and motivating that you did that.
Thank you.
Yes,
So
থাকার পরেও অনেক সুন্দর করে বলছেন, এটা অনেক বড়
সেটাই,
একটা ব্যাপার।
it is
মাঝখান
খুব
দিয়ে
ভালো
মাঝখান
লাগলো.
দিয়ে আপু হচ্ছে কাশতেছে, মানে তাপে বলে
I'm really sorry for that. আমার
No,
আসলে
we can
মাঝে কাশি
understand.
হচ্ছে,
It's
মানে
it's
খুবই বাজে
nice.
অবস্থা।
It's nice that you did that, even though you had trouble in talking. Anyway, we will see you in next presentation, and we encourage
Inshallah.
everyone who haven't, please do present at least for once. It will help you. Believe me, it does. At least when you start presenting, I mean, if you start once, it's like a stage fright. If you have any stage fright, someone who know how to act, know how to perform, but they're afraid to get up on the stage and start to move. So it's like if you have anything like that, you can break that boundary, that what I can call, that timidness, whatever. So thank you everyone, and enjoyed it. But I really we all know, but I I myself don't know all of the terms, all of the experiments sheet that was included in this paper, but it was an interesting one. And yeah, so anyway, and I'm talking too much. uh, Good night everyone, and see you next presentation.
Bye, good night.
জি,
Good night.
good night.